everyone. As I mentioned in our previous lectures for Chapter 3, I would love for us to do a transcription and translation example. In order to do that, we need a strand of DNA and then utilize that information to convert it into RNA. And once we have RNA, we will use our codon tables to figure out the specific amino acids needed to create our polypeptide, our protein strand. So let's go ahead and take an example of a DNA strand. Now, the reason that you know that this is DNA is because you notice that it has the T for thymine. Remember, that's exclusively found in DNA. And I also want to remind you that not all of your DNA we can utilize for transcription. It will only be the genomic section, so the parts that we call a gene. So keep in mind that a gene, by definition, is a strand of DNA that you're able to do transcription on. Now, since DNA is housed in our nucleus and it cannot leave the nucleus, what we see is that any time that we need information from a gene, our first step will be to copy the DNA and convert it into RNA. And when we're doing that, we are doing transcription. So our first step is to look at our strand of DNA and go ahead and use complementary base pairing to figure out its RNA strand. So keep in mind, G will complementary bind with C, guanine and cytosine. Adenine will complementary bind with uracil because there is no T or thymine in RNA. So the A will bind with a U. However, if you come across a T in DNA, that will still complementary bind with the A for adenine because RNA has A, it has a G, it has a C. The only difference is that instead of a T, you will have a U. Now, I would recommend that you pause the video at this point and go ahead and give yourself ample time to go through the DNA strand and come up with the complementary sequence. So I'll start you off. The G will be matched with a C. The A will be matched with a U. And go ahead, go down the entire length of this DNA strand. Take your time and match it up with its complementary RNA strand. When you get to the end of the sequence, it should look something like this. So here in blue, we see the end result of our transcription, which is to generate a complementary RNA strand based on the gene or the DNA sequence that we were provided. Now at this part, what the cell would then do is it would do the splicing and it would get rid of the introns and keep the exons. And of course, it would stabilize it by providing it a poly A tail on the 3' and on the 5', it would provide it with a cap. So we are going to safely assume that the sequence that we're looking at has been fully stabilized and it is ready to go. So our RNA will move or migrate from the nucleus into the cytoplasm, where we can then continue our step. And what we want to keep in mind at this point is that when you do your translation, so take your mRNA and start producing your proteins, that we won't read the RNA strand in its entirety. Instead, we're going to read it in sections of 3 nucleotides, aka we're going to read it as codons. So what we need to do is divide up our strand into codons. And honestly, the easiest way to do that is to just kind of section off 3 at a time. And as you're doing that, your final strand starts to look a little bit like this. So here you go. You can see the division of the codons, 3 nucleotides represented in our mRNA. Now, as we discussed in our lecture, when you do transcription and translation, it occurs in 3 events. You have your initiation, your elongation, and your termination. The initiation step of translation requires us to locate start codon. Now, of course, we can reference the codon table for that, but we should know what the start codon is by heart. And the reason for that is because it's a universal codon. All organisms that do translation require the same 3 letters to get it started. And that is going to be AUG. I hope you came up with that as well. Now, when you do your translation, you always want to start with AUG. That is going to be the start codon. That is going to allow the rRNA to recruit the larger subunit onto the smaller subunit and start the process of collecting codons and matching it with the amino acids that the tRNA is going to bring over. We are not concerned with anything that happens before the start codon, so you can just ignore that. And what we're going to do now is go ahead and start exploring the codons that come afterwards. And we're going to utilize our codon table to determine which amino acid it aligns with. So here is our codon table, and our first codon that we're going to explore is what is GGC going to bring us. And keep in mind, in the cell, that would be the tRNA that would be going over and getting the particular amino acid. Here's our codon table, and we're looking for GGC. You can explore the table however you'd like, but I think the easiest way to do is to follow the directions that the table provides us, which says that the first letter should start on the side. So we're looking for GGC. So let's find our G. Here we go. And then for the second letter, we're going to go across. Okay? So we found our G, and then we're going to find our second letter. That's also a G. So that will bring us into this particular box right here. And the C is the last one. So we're looking for glycine. And you can indicate it as glycine or GLY, whichever one you remember. That's perfectly fine. So let's go ahead and go back to our sequence. Let's report that we found glycine. All right, let's keep this going. So the next codon is going to be CGU. Back to the codon we go. So we have our C. We know our G is over here. So that's going to bring us right over here to this box. And then we have our CGU, so that's arginine. So let's report that one back. All right. And ACC is going to be our third one we're looking for. A, there you go. And then C is right over here. So that brings us to the box. And ACC is going to give us proline. All right. So far, we have collected 3 amino acids. And keep in mind, when the tRNA brings over the amino acid, the tRNA has an anticodon that is complementary to the codon of the mRNA to make sure that it's putting the amino acid in the proper location. All right, next up is UAA. U and A. All right, that brings us to this box right here, UAA. Oh, that's a stop codon. All right, it seems like we've reached termination, which means that we're about to end our translation step. Let's go back to our sequence and report that. So I went ahead and I wrote down stop on mine. So now I have my starting point. I have my ending point, which means that these are my 3 amino acids that I'm interested in building my protein. So what I always like to tell my students to do is once they have the start and the stop, then they want to go ahead and just mark the fact that they don't care about anything before the start and they don't care about anything after the stop. We simply care about the amino acids that are in between. It's always helpful to clearly label your final protein strand. Now, from a personal preference, I like to mark the amino acids and kind of get rid of the start and the stop. However, some people will write down start and stop, or some people will write down augine or methionine, which is the amino acid that it brings in case. It really is the preference that you're looking for. But at the end of the day, you want to keep in mind that if we're following the rules of initiation, elongation, and termination, we're only caring about the codon sequences that happen between the start and the stop. Now, I should also put a little disclaimer, which is that in the cell, these sequences will be a lot longer. And the RNA polymerase and the tRNA, rRNA, and mRNA will move a lot faster, but that's okay. Either way, we were able to display an example where we started off with DNA and we worked our way and made RNA out of it. And from that RNA, we went ahead and read the codons to come up with our peptide strand, the start of our protein. I hope that was helpful, and I will see you soon for our next chapter.